Okay, thanks to take a seat. We're going to start last session, session 4, dedicated to cell therapy and beyond. And this afternoon, no round table, but a succession of short talks. Then, of course, time for questions and answers. Two very important keynotes with Professor Philippe Menasché at first. And, of course, Professor Alain Fischer. It will be also the occasion, we'll get some time for questions and answers for them too. And to start, Mr. Luis Casteilla, you're going to talk about MSC-based ATMP, present and future, and you come from Toulouse.
Thank you. May I have first slide? No. Sorry. How does it work? This one? Ah, yeah. Okay. Hello, everybody. It's a pleasure for me to present this talk, and it's a challenge in a few minutes to present a large overview of MSC-based therapies, and also I would like to share with you two achievements from my lab. So, first of all, to understand the role and the function of MSC, it's necessary to take a step back. Indeed, any function in any organ is a result of a strong interaction between two cell compartments. The first one is a parenchyma, which contains the specialized cells, such as cardiomyocytes for heart, neurons for brain, and stroma. Stroma is a ground for the specific compartment of parenchyma. And with such picture, you see that MSC can be found in both compartments. But on one end, in the parenchyma, they can behave as progenitors for specialized cells, for adipocytes, for cartilage, for osteoblasts, for tenocytes. And this behavior is only in mesenchymal tissues. On the other hand, you can find mesenchymal stem cells in all tissues, in all stroma, such as a network which is responsible to generate, to structure, and to stabilize the stroma. And so why and how MSC are able to do this job? Because in fact, they have many functions. Of course, they are multipotent. The name is stem or stroma cells, but also they display a great paracrine activity. And through these different activities, they are able to manage a lot of things into the stroma and into all tissues. The first point is they are able to manage the inflammation and the immunity with a capability to interact with all immune cells. They are also able to manage vascularization through the secretion of angiogenic factors, but also through their capability to behave as pericyte and to stabilize vasculature. They play also a great role in the structure because, in fact, they give rise fibroblasts. They are responsible to secrete extracellular matrix, and they are involved in their remodeling. And finally, also, they have a great capability to handle the metabolism through their capability to differentiate into adipocytes. So, through all of these functions, they behave as a true conductor for all organs. And so, with such picture in the mind, if you think about MSC-based therapies, it's necessary to consider two quite different paradigms. The first one, of course, is to use MSC as progenitors for specialized cells. It's a first option. The second option is to use them to indirectly recover or to indirectly correct some tissue dysfunction or some function impairment. And so you can use MSC for their role as supportive function, and a role as conductor for any tissue. And in fact, if you try to follow, to make a picture of the clinical trials in the world, you see that most of clinical trials in the world in the field of MSC are clinical trials devoted to the function of the supportive function. And you find different clinical trials involved in quite a large number of applications and disease, and you see here the use of MSC as mesenchymal precursors, and you see that the number of clinical trials is relatively limited. And if you go through the literature, it's clear that MSC are very effective, for instance, in GVHD. In Crohn's disease, you have a market authorization in this field. You have promising results in intervertebral disc and also in critical limb ischemia. So all context corresponding to the context of supportive function. So it's a picture of MSC. Now, how we can move forward? In fact, the results obtained with MSC are relatively variable. And so it's necessary to try to have robust effect. And so to do that, we think about the fact to generate a therapeutic 3D mesenchyma or a therapeutic 3D stroma in order to correct the dysfunction in the tissue. And to make the story short, we made a proof of concept, in fact, in an emblematic disease, which is an age-related chronic inflammatory disease associated with dysbiosis, which is periodontitis. And in fact, we set up a clinical trial in aged dog that display, aged dog are very relevant model because they display exactly the same symptoms and the same feature than the disease in human. And using aged dog and using a strong design, which is a randomized double-blind control trial, comparable to a human trial, we were able to demonstrate that if you bring ASC, the network with hydrogel, we are able to improve inflammation with a decreased bleeding. You improve the growth of the tissue, and even you correct the dysbiosis. The correction of dysbiosis perhaps is not permanent. We are investigating this point now. So you see that if you are able to bring ASC in the correct form, you can have a strong result. The second achievement I would like to share with you is a different achievement. It concerns the use of MSC as progenitor cells. And to do what? To do adipocytes. And you can ask, adipocyte, what is the interest? In fact, in the field of metabolic disease, there is a very interesting adipocyte. The name is beige or brown adipocytes, because such cells is able to burn energy, and in fact, it's a way to limit the energy storage and to limit obesity and metabolic disease. And again, to make a story short, here, in fact, we set up a bioprocess suitable to produce organoids with our very relevant model to screen all drugs you want, but using the same process and by scaling up, we are able to generate a 5 millimeter object, vascularized, which is suitable to be transplanted in order to correct any metabolic disease. So now, presently, we are investigating the result or the consequence of the transplantation of such object in obese or diabetic models. And so, just to keep the time, I think I was perhaps too short, but here you have the conclusion. I think MSC, this is not the end. We have great hope in MSC. Okay, it's different from CAR-T, but you have very challenging field, and okay, we believe in this field. But it's necessary to have a new understanding of MSC. It's necessary to have a new look on MSC and the fact to use MSC. How we think about MSC, I hope that I convince you that 3D models are very relevant, and it's easy, relatively easy to get and easy to manipulate. And of course, I don't speak about exosome and engineering MSC, which is another topic, and the time was too short to speak about this. But this domain is also very active. So, thank you.
Thank you very much. Thank you. And of course, you'll come back and we'll talk. I don't know if you hear me. Hello, hello. You'll come back for questions and answers. Now, Christelle Monville for a second talk about stem cell therapy pipeline, focusing on retinitis pigmentosa and other rare indications with high unmet medical needs.
Thank you very much, and I would like to thank the organizers for the invitation. I'm really glad to be here. And if I could have the presentation. Okay, so yes, I will talk about retinitis pigmentosa and more largely about retina and the programs on cell therapy that we have developed in the lab. And just to give you a brief insight on what we're talking about. So the retina is the photosensitive layer at the back of the eye. Is a very, very organized layer, as you could see here, with many cells that has to be put in the right order and right direction. And among those cells, the cells that are very important for the sight is the photoreceptors, the cone and the rods that are just there. And at the very back of the eye here, you have a layer of retinal pigment epithelial cells. And those cells are really the babysitters of the photoreceptors. So they provide nutrients, they clean the photoreceptors, they are very supportive cells. And actually in some disease like retinitis pigmentosa, in about 5% of retinitis pigmentosa, when those cells are dysfunctioning, not providing the right function, then the photoreceptors will die. And it's also the case in the much more frequent disease, which is the age-related macular degeneration, the AMD, wet and dry form, where the same cells are dysfunctioning, eventually dying, and then the photoreceptors will die, and then the patient loses sight. So the idea we developed in the lab was actually to differentiate some RP cells from embryonic stem cells, from pluripotent stem cells. We do that with human pluripotent stem cells, embryonic and iPS. And then to generate a little patch, because you saw that in the retina, RP cells are very organized. They are forming a layer. So we generate a patch to reconstruct this layer. And the idea in the first program that we actually developed was really to delay or even to stop the degeneration of photoreceptor. So we developed this product, and because I don't have much time, I will have to cut a very long story very short. So we developed this little patch that you could see here, which is a SIM card format to keep the orientation of the cells. The patch is about 5 to 3 millimeters, and it's embedded in gelatin to actually keep also to protect the cells, to give some elasticity to the patch, to be able to inject the patch under the retina. And we also developed some tools and some specific medical device to prepare this patch without using scissors and forceps, and to provide like a clé en main this patch to the surgeon. So we did that in close collaboration with the Institut de la Vision in Paris, with the Hôpital 1520, Hôpital Saint-Louis, and the AFM Téléthon. And actually, we perform all the steps to prepare, so it's more than 10 years of work. So we did all the steps, so by concepting and having the right protocol to obtain the right cells, so really work on quality controls, performing new quality controls and improve the quality controls, but also we did all the preclinical animal validations needed. I talked about this medical device, and we started actually the clinical trial in 2019. It's a phase 1/2 clinical trial. So the first point is really to tackle the safety of this product, so to look at tolerability, safety of this product. And actually we were able, despite of the pandemic and the COVID-19, we were able to graft 7 patients that are followed in the Hôpital 1520. And the PI, the surgeon, was the Dr. Stéphane Bertin. And those patients have actually specific mutations, but they have 2 different mutations. So it's a cohort of different patients that we followed in this first clinical trial. So where are we now? So we're following this patient, but in the same time, we are trying to improve this patch by trying to automate the scale-up of RP production in order to go to monogenic disease, rare disease, to eventually a more frequent disease, the AMD. The AMD is about 2 million patients just in France. So it's a lot of patients. So we worked a lot on automation, and we have validated actually the protocol that allow us to produce the RP in a different automate. And from 1 million, we obtained 16 billions of cells with exactly the same quality than the one that was done manually. And the second aspect is also to work on cryopreservation, because we know how to cryopreserve the cells, we know how to cryopreserve the membrane, but now we're trying to cryopreserve actually the entire patch to have a sort of a off-the-shelf product that would be easily to go to the patient, and to also reduce the cost of the production of this patch. And finally, so what are the perspectives beyond this RP patch? Of course, is to add the other types of the cells that are in the retina, so adding the photoreceptors on the patch. So it's a work that we are performing with, in collaboration with Institut de la Vision, and also with some physicists in Paris-Saclay, because we need matrix, we need scaffold to put the cells together and to do some tissue engineering, to have more complex tissues to actually cover the entire spectrum of retinal dystrophies. We're also working a lot on trying to escape immune system, so working on getting more some immunotolerant cells by different approaches. Working also combining eventually extracellular vesicle therapeutics to maybe decrease the inflammation, prepare the host environment, and also trying to improve the quality control by using some artificial intelligence. And just to finish, this is the pipeline that we are addressing. So we have a strong pipeline addressing different types and stages of all the retinal diseases. So in monogenic disease, so in retinitis pigmentosa, the patch we have developed cover about 5% of the disease. So we're working on trying to develop products for the other 95%, but also developing a more complex patch for more frequent disease like the AMD at different stages. And just the last slide, but maybe the most important, is to acknowledge all the people that have worked since more than 15 years now on this program, and also the patients that are participating to the clinical trials. Thank you.
Thank you very much, Christelle. And now another short talk, Frederic Desdouits, and you're going to talk about unlocking cell therapy for all, and you're presenting Treefrog Therapeutics.
Good afternoon, and thank you very much for inviting us. As I have just a few minutes to present what we're doing, this is a bit difficult. I have only one slide, so I'll try to make it very simple. And this slide, you'll see, contains only numbers. So if you can move to the next, or the first, the only slide. You want me to? Okay. Okay, so Treefrog is a cell therapy company. We're based just outside of Bordeaux, in Pessac. And I'm Frederic, I'm the CEO. Okay, just a few numbers. So hundreds of billions, that's one of the challenges of this industry, is to be able to make batches, to manufacture cells at this scale. If we want to go for large organs, there will be a need for very large manufacturing scales of human cells. Nobody can do that today. What we heard in the previous presentation, 15 billion, 20 billion of cells is about the limit of what people can do today in terms of, let's say, solid organs. I'm not talking about immune cells. 120, this is the number of froggies we have. So we are 120 people, a bit more than 100 here in Pessac, and we have now a lab also in Boston. We've been very lucky because we have been able to attract a lot of very talented people. We have a very large team now in the field of cell therapy, and we are in the cell therapy world focusing more even on regenerative medicine, so solid organs again. So we're, pretty large company for a French biotech, pretty small when we compare to the field and the competitors. We do a lot of things. We do process development, obviously, a bit of manufacturing, but we do also biology, obviously, but technology, and we are a technology-driven company. So we've raised $82 million so far, so the 82 you see here. So what we've done with this money, we built an organization, as you see, but we also develop our technology to a GMP status to be able to provide a solution for our partners and also for our internal programs. We have two large programs ongoing, one in Parkinson's disease, where we think we have a best-in-class approach based on microtissues with neurons. We are the only company in the world to graft neurons. And also a program in liver, in acute liver failure, where we intend to provide the patients with artificial livers, small livers, because hundreds of billions of cells, even if we can produce them, is just a few dozens of grams anyway. It's not huge. There are three challenges in this industry. One is scale, as I said, obviously, and our technology is scalable. That's one of the rare technologies that is scalable today in this field. The second one is the quality of cells. So this morning, Karine talked about it. We are injecting cells into people, and the quality of cells is super important. Cell therapy is about cells. And when you start to reach those levels of mass production of cells, most of the time, the quality of the cells is a question. So fortunately enough, our technology brings, let's say, a 3D biomimetic environment to the cells. And so far, what we've seen is that it prevents, let's say, damage to the genomic integrity of the cells. But still, it's a very important focus of the work we're doing. Third important point in the field is the functionality of what we're doing. When we change scale, when we move from the bench to the bioreactor, it's very difficult to keep the same functionalities, to keep the same quality of cells. It's a big challenge. And again, we think we have solved and we have tackled this challenge. The next number 3 is the times we think we have made firsts, world firsts. We were the first to build a high-throughput encapsulation system for human cells. So our technology is simple. We are creating bubbles of alginate around stem cells that prevent them from any shear stress in the bioreactor. When they are in this bubble, we can amplify them, we can differentiate them in any type of microtissue we want. So the first machine that is available today as a GMP, with a GMP status. Second first premiere is that 3 years ago, it's already 3 years ago, I checked because I had a question yesterday, so it was in 21. We made the first bioreactor of stem cells that was large enough to produce in 1 week 15 billion of stem cells in one bioreactor. So it was 3 years ago. And I guess that's still the limit of what the industry can do. We are already working on the next generation of this technology to be able to reach the number you see here, hundreds of billions of cells per week. And the third first premiere or first world first we did was that we were the first company in the world to inject mature neurons in animals. It's very difficult to manipulate neurons, as you know, and the way we manufacture our microtissues, that we have 3D objects in which that we can mature enough, where we have mature neurons, and we can do the same with hepatocytes, as you can guess, but with cardiomyocytes and any type of cells, because we can protect them for a long time, so we can keep them protected for weeks. And we are the only company still in the world working on injecting mature neurons in the brain of animals so far, and soon in humans. And I will finish my talk with number one. We have one purpose at Treefrog. It's cell therapy for all. So we are developing our own programs. We're also working a lot with partners to help them to break the glass ceiling of cell therapy, which is basically translational research. I mean, most of the companies are stuck in preclinical stage because it's very difficult to move to the clinical stage at the right scale with the right product. It's not just the scale, it's the right scale with the right product. And that's the aim of the company, the purpose of the company, cell therapy for all. With that, I'll thank you, and we'll go on, I guess, with questions later on. Thank you.
Thank you.
And maybe just to finish up, just to show you the team in Pessac, so a lot of good people. Thank you.
Thank you. Great. Thank you very much. You will come back in a few minutes for questions. And to end this first session, this part of the session, we'll have a video because unfortunately Christian Jorgensen couldn't be present in flesh today. So we sent a video to talk about next generation of cell therapies for osteoarticular diseases.
For cure in Montpellier, and I will take the few minutes that we have to have an update on mesenchymal stem cell therapy. So a cell therapy dedicated to osteoarticular pathology. So first, very briefly, what is the unmet medical need? As you know, there are an increasing numbers of patients in Europe and in the US with osteoarthritis and vertebral discopathy and degenerative articular disease. And today there are some options, including options in orthobiology, using PRP, using a combination of hyaluronic acid and PRP and others, and we also have the option to develop cell therapy. Can be a chondrocyte-based therapy or stem cell-based therapy, and there have been in the past years a lot of initiative and clinical trials, and I will share with you some of them. Just to know that in Korea, one of these products has been approved for osteoarthritis. And in Europe, Alofisel has been approved, but in fistula, in gastrointestinal pathology. And so very, very briefly, mesenchymal cells are interesting because they can differentiate to tissue that are supporting tissue, including bone, cartilage, muscle, fat tissue. But they also have paracrine activities, and they release EVs, but also cytokine factors, and globally they reduce inflammation. And so when we want to translate that in the clinic, as we have done in some of the program, the first question is, should we go to an autologous process or to an allogenic process? The first initiative was using autologous cells, because then you can re-administer the cells. There is no problem with safety. But one of the problems is the cost, because that means that you have to, for each batch of cell production, you have to have all the quality controls, and so this is increasing a lot the cost and the disponibility. And of course, an allogenic cell program, when you have cell banking and then you have access immediately for the physician or the orthopedist to the cell product that makes things much easier. So very, very briefly, we have to organize either the bone marrow or the fat tissue aspiration, and then you go to a GMP facility, you expand the cells, and you can cryopreserve the cells, and when you need, you have access to the cell product. You have a control of viability, you have a control of cell quality on the phenotype, and also on functional assays, and then it comes to the physician room, and you can be injected. So one example is osteoarthritis, a knee joint osteoarthritis. So you have local injection, and what you can see is you have some improvement in terms of pain. You have some improvement in terms of function. That's the Coos analysis. But it is a modest improvement, and other and recent papers have shown that you have some improvement, but it is limited when you look at 12 months. It's limited. It's improvement between 20 and 30%. So there are some limitation. And another very good indication is the intervertebral disc. And so as you see here, you have a decrease in the proteoglycan content and the hydric content, and you can inject using scanography with the needle. You can inject directly 20 million cells in the disc, and when you do that, you are improving lumbar pain for the patient, which is something important, and you can see here, and we can also see that we are improving the hydric content in the disc. When you do MRI at months 12, months 24, we have some improvement in the imaging. But however, there's clearly a need to improve the biological effect of these cell products, and one of the way to improve is to impact the genetic program of the cells, and we can do that with inhibitors of PPAR gamma. This is one example how you can do that. You can, when you do that, improve not only the immunosuppressive effect, but also in vivo, and you are improving the potential of this cell product. Another way is to use vesicles that are expressed by the mesenchymal cells. So you have a mesenchymal cell lines in your bioreactor, and you can obtain all the extracellular vesicle that you can centrifuge, purify, and you can use this as a biological product, but it is not a cell product. It's very safe. You don't have any DNA. And so this is a way to improve. And at last, you can also obtain skeletal progenitors derived from iPS cells with a clearly improved of the cell product to obtain a new cartilage that can be formed, implemented in some in vivo models of cartilage defects. And with this, clearly obtain a fetal cartilage, but now there's some step to prove that this is functional, that this is biomechanical relevant for adult tissue. So this is an area where the unmet need is very important. There's a market access very important here for developing a cell therapy program. MSCs seems very safe, but the improvement is today modest, but clearly we are improving with the next generation of cells, genetically modified, or using EVs produced, or using MSCs derived from iPS. So there's a lot a new product coming on actually in development in early phase trials. And so with this, if you have any questions, I am absolutely available to reply. You can contact me by mail. That is here on the screen. And again, I apologize not being with you today, but I think you have a very good meeting, and let me know if you have any questions. Thank you very much for your attention.
Thank you for him. So I'm calling back now Louis, Casteilla, Christelle Monville, and Frederic Desdouits, so as to have a session, question and answer session. Come on stage, thank you. Take a seat wherever you want. Yes, I don't sit, no, I don't. No seat for me, no. Forbidden. Over there, wherever you want. Yes, there. It's okay. You want to be close to me? Yeah, I can see. Okay, so do you have a first question? Who wants to start first? Okay, yes. Professor, of course. Alain Fischer. Alors, push on the button, le bouton.
Merci.
Voilà.
Okay.
Thank you.
I know very little about mesenchymal stem cells. Still, I will try. It seems to me that one of the problems you may face for the future for clinical development is reproducibility of results of batches of cells. So how can you say improve or develop methods, I would say markers, biomarkers of the quality of the products and the reproducibility of the quality of the products?
Right? Who wants to answer? Yes, Luis Casteilla.
By working. Because, of course, no, at the beginning, the definition of these cells was very simple and too simple. And so it's not a simple phenotype with some antigens, which is able to tell us what are their properties. So we have to deep into the cell and using transcriptomics, etc., etc. But I think the best way to qualify such cells is their answer to a challenge. So we have to qualify the cells by using a test, a challenge test, because the true answer is there, and not basal and basic definition.
If I can
Yes,
add to that, because obviously we have an approach which is very industrial and manufacturing, let's say, manufacturer-oriented. In our case, we have, out of the 120 people, we have 10 people in analytical development, so just analyzing what we're doing. We have a team of 6 people working only on iPSC quality. So what is an iPSC? When you start to use an iPSC, you know, after a few cycles, what's happening in the box, or whatever you're doing. We have a team of 4 people in imaging, because everything we do imaging. We look at the cells, just, you know, you need to look at the cells. And actually, the discussion we have with EMA and FDA now, they are requiring now immunofluorescence on almost every product we produce, just to make sure that the cells we have are the cells we think we have. So it's a huge work. But to get there, what we do is that we have developed a completely closed system, which means that once the cells are in the bioreactor, they stay in the bioreactor for weeks, so nobody's touching them. And you can change the media, obviously, but there is no human interaction with the cells. There is no open system to protect the cells, to have something consistent, reliable. And we run a lot of manufacturing batches. So today, for instance, the Parkinson's program, we run a batch every other week. So we start to have a lot of data, and we know our product. So that's what we call manufacturing. It's we are there.
Okay,
we're getting there.
Yeah.
I don't know if we're getting there.
And in the lab, we used automation, just not for scaling up, but also to have a large batch of cells. And automation allows us more reproducibility than human practicing. And also the idea is to have a qualified bank, and you have one vial, one experiment. So qualification, and it's probably more homogeneous. It's not the case for you can't do that for every cell, but we're working with iPS and human pluripotent stem cells. And basically we've been able to do that with pluripotent stem cells, but also with differentiated cells. So I think also it's a way to kind of homogenize a little bit the cells and decrease the differences batch to batch.
I think another issue is the selection of the donor, because in fact there are very few works in this field, and the selection of the donor is poorly documented.
Thank you. Another question? Yes, Marc.
Yes, along this line, what is not completely clear to me is how you can formally distinguish an effect linked to regeneration versus supportive function. Because in many instances, basically owing to, for instance, the anti-inflammatory properties of MSCs, they might trigger actually an endogenous regenerative process, but not mediate themselves this regeneration. So I don't know whether this is a relevant question. No?
Very good question.
Yeah. In fact, my belief is that regeneration is possible even in adult individual. So we have just to unlock some inhibition or to manage some stages of the regeneration to promote endogenous regeneration. So my view for MSC is that MSC has a good tool to do that, to correctly manage inflammation and immunity in order to promote endogenous regeneration. So it's true that you have a mix, of course, between both.
Right. Another question? No more question? Yes, sorry.
Just to comment for the last question. So I think the MSCs, the cells cannot contribute in the regeneration without scaffold. For the retina example, for example, they have some scaffold because we need some matrix. We need the adhesion of these stem cells to support to start regeneration. But MSCs in solution, like injection, can decrease the inflammation, for sure, the pain also, but cannot regenerate if we don't have this attachment to a scaffold or a support for this regeneration.
No comment?
I think it's a lot depending on the application. I think for the retina, you really need this organization because the cells are forming layers. But people have tried, all the clinical trials have tried to put the cells into suspension and try to, say that the cells will find their way and they will organize by themselves in the host tissue. Sometimes it happens, but it's not complete. And for those cells to have the right function, they need to be organized and they need to be polarized. For the MSC, it's probably a little bit different and it's probably depending on the application.
Sure, they depend of the application, but we cannot see a regeneration in this case. We will have some secretome, we will have some modification, some reparation, but not regeneration.
But where I agree is about the formulation. I think the formulation is really important.
Yeah, for sure. Yeah.
Yeah, it's why we propose 3D therapeutic mesenchyma.
Yeah, we're not working on MSCs, so I'm not an expert on. I'm an expert on anything, but not on MSCs for sure. But our experience is that when we encapsulate cells, though they are in like those small bioreactors of a biomimetic environment, clearly you're right. I mean, they need a structure. All cells need a structure. They need something they feel comfortable in. So MSCs. But it's true also for T cells, it's true for NK cells. Even the cells we think are circulating, I mean, they need structure. They don't like to be floating around.
Thank you. Another question? Yes, Professor.
This is for Christelle Monville. Could you summarize what are the advantages and disadvantages of retinal stem cell therapy versus gene therapy for monogenic diseases?
Well, I think it's, again, it's about what disease you're tackling, what stage, and I think they could be complementary. I think. They're not in opposition. The stem cell strategy is probably you could go for a different mutations, and that's what we want to actually show. Is the clinical trial was performed in patients who have different types of mutation, whereas gene therapy, when you tackle one mutation, it's mutation driven. But you have gene therapy for the retina where you put trophic factors, and this could be for different type of retinitis pigmentosa. And also the final gene therapy for retina is the optogenetic clinical trial where, again, it's mutation free. So it's probably depending on, again, what type of patients, what the stage of the disease, and probably for some patients, stem cell therapy will be a better approach, and for other gene therapy could be good. So it's not in opposition, but it's for complement.
Thank you. No more questions? Yes.
Yes, I have a question concerning the scaffold of your application in retina. So you have, what is the origin and the composition of your scaffold?
It's a human amniotic membrane.
Okay. Do you need to have because it's a medical device, right?
It's
and
not.
the combination with cells.
It's not a medical device.
Okay. It's a combined scaffold and cells.
So it's a supportive scaffold. So it's a sort of it's not a medical device. It's coming with the product, but it's part of the product. So it's part of the IMP. So but it doesn't have an effect. So the effect is given by the cells, but it might provide supporting factors. The name is just not in my mind now, but.
Okay, thank you.
Okay. Yes.
Yes, hello. That's for MSC. I was wondering if using a scaffold would help overcome the viability of patient donor, because we see a lot of difference, for example, for autologous therapies. So the quality of the cells and the phenotypic controls can have different results. So would using a scaffold overcome that problem?
Yes, to provide some support to the cell, in fact, you stabilize. And if you go back in the past, the name for MSC was reticular cells. That means cells in network. So to have a good functioning for such cells, you have to generate this network. And so by providing a support, I think it's the best way.
Okay, thank you very much for your answers. Thank you. And now I'm calling Professor Philippe Menasché. You know him very well. Of course, great pioneer in heart cell therapy. And he will talk about that today. And he's from Paris Cité University, between others. Professor.
Well, thank you. And first of all, I would like to thank once again the organizers for participating in this very, very fruitful meeting. So, just for those of you who may not be quite familiar with heart failure, just a couple of figures to highlight the magnitude of the problem in terms of number of patients, in terms of prognosis, which remains poor, and also obviously in terms of costs. So, to make a long story short, the major underlying cause of heart failure is the loss of millions of cardiomyocytes, contractile cells, leading to a deterioration of heart function. Now, when you have a sick horse, that could be true for any other animal, you have two major options. Option one is to heal the animal, which transposed to the failing heart means to repair. Repair involves different mechanisms that you show here and which can contribute to improve heart function. But you will note that there is nothing to do with regeneration, which would be the generation of new cardiac cells. Anyway, in this context, it makes sense to use either cells or exosomes. Exosomes is a poor word. We should talk about extracellular vesicles at large, not to say secretome. Now, second option is to replace the animal, which transposed to the failing heart means triggering the generation of new cardiomyocytes. And this is the only definition for regeneration. And it is in this context that RNA therapeutics can find their place. So I think these are two major conceptual options which should be kept in mind when you're thinking in terms of therapeutic applications. Now, a brief snapshot on the landscape of cell therapy. Currently, there are two major types of cells which have been and still are investigated clinically. One hand, mesenchymal stromal cells, and there are previous talks which have addressed issues associated with these cells. And in the context of heart failure, we should acknowledge that the data so far have been conflicting. Some trials reporting positive outcomes, others negative outcomes. Second category of cells, cardiomyocytes, derived from pluripotent stem cells, either ES or IPS. Now, it's too early to draw conclusions regarding efficacy because these trials are on the way. However, in terms of safety, it's rather reassuring. So far, no red flag, and I can tell you that the first patient in whom I transplanted cardiovascular progenitors derived from ESCs is now 9 years from her surgery without any side effects that could be attributed to the cells, and this is also true for the other patients of this trial. Now, something important that we have learned from this accumulated experience with cells is that in contrast to the initial expectations that they would engraft and make new myocardium, actually, at the end of the day, and no matter what you do, so far, they die. Nevertheless, there is still some benefit, and this has led to the so-called paracrine hypothesis, whereby the primary mechanism of action of the transplanted cells is to secrete a blend of biofactors, many of which being packaged in extracellular vesicles, in which harness endogenous repair pathways, the same pathways that I have shown to you, that is, reduction of fibrosis, reduction of inflammation, stimulation of angiogenesis, and so on. So, that said, this equivalence between stem cells and their secretome has now been established in a wide variety of preclinical models spanning, as you can see, very diverse diseases. So basically, the effects of transplanting stem cells can be duplicated by the exclusive delivery of their secretome. This has been a major achievement of these past years, which raises the question, if there is functional equivalence, why would you use exosomes or EVs instead of cells? Well, the answer is that using the secretome features definite clinically relevant advantages that you see here, and probably the most important of these advantages is the lack of immunogenicity. There is a current trend to use allogenic cells, in particular those coming from pluripotent stem cells. Obviously, these cells will trigger an immune response requiring immunosuppression with all the side effects we're aware of. Well, If you take the secretome, in particular of cardiovascular progenitor cells, definitely it's not immunogenic, which represents a major advantage for the patient. This explains why, so far, you really have a bunch of clinical trials ongoing with the use of secretome coming from different cells, largely MSCs, but not exclusively. And now there is another one, which is our secret HF trial that we have started now 6 months ago. And basically, just to give you a brief outline, we start from IPS. The IPS are clinical grade, obviously IPS, are differentiated into clinical grade cardiovascular progenitor cells, and then we collect, purify, filter, concentrate the secretome, which is injected intravenously 3 times, separate by 3 weeks, in patients with non-ischemic cardiomyopathies. We've treated the first patient 6 months ago. Everything has gone well so far, and the second patient will be treated next week. So, cells and exosomes share a couple of issues, some of them being listed here. I will not detail them because I just want to highlight which, for us in the clinics, probably is the major bottleneck. And the major bottleneck is delivery. There are 2 ways of delivering cells or exosomes, and I don't think that so far we've been very successful in managing delivery very well. The first option is to directly deliver the therapeutics, either the cells or the exosomes, in the organ, okay? The advantage is that you definitely hit the target. The problem is, to do that, for the heart, you need an invasive approach, either a surgical open chest approach, which makes me happy, but it's not very easy for the patient, or a catheter-based endocardial approach. But in the 2 situations, you have an invasive route, which means that it's going to be tricky to repeat the procedure, whereas these patients definitely probably need to be redosed from time to time because it's a chronic disease. Now, the second option, which is by far much more attractive, is the IV approach. It's easy, it's non-invasive, it can be repeated, it's not expensive. But then the mechanism is not that easy to understand, and we come to the kiss and fly hypothesis. Kiss and fly meaning that the current hypothesis is that once you have injected IV, either the cells or the secretome, it's basically trapped in the spleen, largely, but primarily in the liver and also in the lungs, where it might shift the phenotype of the endogenous immune cells toward a reparative phenotype, and then these reprogrammed immune cells then traffic in the blood flow and hit the target organ. This is how we can explain the fact, in particular in the brain, but also in the heart, when you inject intravenously, the therapeutics never reach the heart, but nevertheless, you have some benefits. But definitely, these hypotheses need to be further characterized, and this is major practical implications. Now, the second option is to regenerate, really, that is to trigger the production of new cardiomyocytes. And once again, neither the cells nor the exosomes so far have been able to induce a true regeneration. Now, the problem here, where RNA therapeutics might find their place, the problem here is that the regenerative capacity of the heart is very variable depending on the species, and unfortunately, in the human adult heart, which is the only one we're interested in, the regenerative capacity is lost. Indeed, it's not fully true. There is a very slow turnover over life, but not enough to compensate for the loss of cardiomyocytes, which underlie the development of heart failure. But the mechanisms, and this is the important point, the mechanisms are there, as shown by this very unique observation of a neonate who suffered a myocardial infarction at birth and completely recovered without any functional defect, without any residual fibrosis. Unfortunately, the time window is very short, a couple of hours or days after birth. After that, the mechanisms in play for cardiomyocyte proliferation are just shut off. And the big issue is, now that we know the mechanisms, how can we reawake them and trigger again, in a controllable fashion, a proliferation of cardiomyocytes? And here comes the benefits of COVID. Why? Well, because COVID is based on the use of LNPs, encaging mRNA, coding in this particular case for spike, okay? The idea has come, why don't we use this technology, but to put an mRNA coding for one of these factors able to reinduce cardiomyocyte proliferation. And actually, proof of principle has now been brought, in particular by this recent study, where they did exactly that, taking mRNA, coding for a factor known to induce cardiomyocyte proliferation in a large animal model, and showing definitely an improvement in function along with proliferation of new cardiomyocytes and a reduction in infarct size. So we're lucky to have been granted by the European Commission now to leverage this mRNA technology to induce cardiomyocyte proliferation by the appropriate identification of the factors able to induce such proliferation and putting them in appropriately targeted LNPs. Just to mention, however, that the issue is not trivial and that we should make clear distinctions between these technologies applied to vaccines and those applied for, say, regeneration. And again, a major difference and a major issue that we have to tackle is delivery. Because here, you have this issue of delivery at two levels. First, if you inject intravenously the LNPs, they have to target the heart and bypass the liver, which is very difficult to achieve. And number two, once they are in the heart, you have to get the expression specifically in cardiomyocytes and not in the other cell types. So you see the challenges of delivery are really there, and one of the objectives of the consortium I just showed is to try to address these bottlenecks. If we look at the landscape of RNA therapeutics right now, most of the studies actually are using microRNAs as biomarkers. There is a single clinical investigational interventional trial testing an ASO for reducing remodeling, which is enlargement of the ventricle, but not really regeneration. So to conclude, the title of the presentation was Cell Exosome RNA. I don't have the answer, obviously. But as you have seen, there are different criteria which can be taken into consideration. First is the objective, repair versus regeneration, and they're not synonymous. Number two, obviously the soundness of the preclinical data, but also, and maybe more importantly, the manufacturing considerations. Because if we don't appropriately address these manufacturing considerations from the very beginning and in conjunction with the regulatory authorities, it's unrealistic to figure out that the basic concept will be ever moved towards clinical applications and patient care, which at the end is obviously our only objective. Thank you very much.
Do you take some questions? Yes? Thank you very much, Professor. Do you have any question for Professor Menasché about his presentation? Yes, Professor Fischer.
Thank you, Philippe. First question. The secretome, that looks interesting, but I would be curious to know what are the molecules that are present, and even more important, among the many molecules that are present, which ones are the relevant ones to regenerate the heart cells? And could it be that based on such studies, you could design a more focused study where we use a limited number of molecules?
Okay, so easy to answer the first one. Obviously, for regulatory reasons, we had to map the full cargo, so we know exactly the type and the number of proteins on board. I can tell you we have more than 2,000 proteins on board, and we also know the microRNAs. So that's just, you know, the list of the components. Now, second question I cannot answer because the fact that a protein or a microRNA is overexpressed doesn't necessarily mean that it's the most bioactive, okay? And so when you look at the literature, it's interesting to see that each study highlights one protein or one microRNA, and it's never the same. So I'm not sure that you will be able to funnel the composition to a very limited number of ingredients which could then make a synthetic drug, because this is obviously the hope of the pharmas. So it might be the case, maybe in artificial intelligence will allow that, but so far the number of components is so huge, and the relations between all these components are so complex, the matrix is so complex, that so far nobody has really been able to say that this is the component or the group of components which account for the protective effects of the whole secretome.
So you mentioned cells, exosomes, and RNAs, not DNA. So I have a question about DNA. I know the guy Erik Olson in Dallas, who has published recently a beautiful paper where he's using base editing to modify the calmodulin kinase, I don't remember which one, in cardiomyocyte, and apparently in animal models, of course, it has a beneficial effect to control heart failure. So I wonder whether you have an opinion on this alternative strategy.
Okay, well, I'm not an expert in DNA, and I just, this was the title I had RNA, so I just talk about RNA.
The criticism is just...
No. This is another option. Obviously, it adds an additional layer of complexity because then you're starting to play with the genome, okay, which is not the case, obviously, with mRNAs. But it's true that there are a couple of studies now showing that some modifications of the DNA and of the genome of the cells might trigger regeneration. My very humble opinion is that we're pretty far from clinical applications in this context. In contrast to mRNA, because with mRNA you have the bricks already there. You have the LNPs, you know how to synthesize mRNAs. And if we can address the very crucial issue of targeting, then we're not that far from clinical applications, I think.
Other questions? Yes, Marc Bonneville.
I perhaps missed something, but when you say, for instance, we are not so far, when using RNA, would be RNA coding for what?
RNA?
Coding for what?
Oh, okay. So this is not the most difficult because the factors known to induce cardiomyocyte proliferation are pretty well known.
Okay. So
And so, for example, within our consortium, the European consortium, we already have the candidates.
Okay. So it would be true.
Thank you very much. Thank you. Thank you very much, Professor. Okay, so another set of short talks now with Grégory Lavieux at first, extracellular vesicle-based vector for targeted therapeutics delivery from INSERM.
Good afternoon. And so first, thank you to the organizer for the invitation. It's my pleasure to share with you
or more recent effort to develop EV-based vector for drug-targeted delivery. So as you heard before, extracellular vesicles are natural vector for intercellular communication. And for a long time, they have been thought to be a very promising new class of vector for drug delivery. But the developments of such a type of vectors has been limited and delayed, mostly because we didn't know enough about how the machinery and how the cell work to really uptake the EVs and how the EV could deliver their content inside the acceptor cells. And I believe that my basic research laboratory contributed to fill this gap. So over the last 5 years, we basically characterize at the cellular, but also know at the molecular level how EVs can deliver their content inside the cells. And I think we still partially a long time debate. So very briefly, the conclusion is that we formally demonstrated that EV can deliver their content inside the cell, but it's a low-yield bioprocess. And so therefore, our conclusion when we decided to contribute and participate to the development of EV-based vector, we concluded that the first task that needs to be done is to improve the efficiency of the EVs in terms of delivery capacity. And for us, the simplest way to do that was to control 3 important steps in the bioprocess: the loading of the cargo inside the vesicles, the targeting of the vesicles towards the cell of interest, and the final step, which is increasing the fusion between the membrane to increase the delivery capacity. So that's what we developed. It has been published, patented, and highlighted already by INSERM. And what I would like to do now in a few minutes is just give you the key results for each modular modules. So for the loading system, we took advantage of a well-established drug-inducible bioprocess, which enabled to force the loading of any peptide-biased cargo inside the vesicles. And as you can see on the graph on the right part, so when we introduce the dimerizer, we can very efficiently load any type of protein inside the vesicle. So the second module is what we call the fusion system to enhance the delivery capacity. And here, we basically repurpose a human physogen called syncytin-1 that is normally involved in placenta formation. And by doing that, we could largely increase the delivery efficiency and reach, as you can see on the graph, same efficiencies that viral proteins that usually are used, but introduce many safety issues. So in other words, we could compete with viral components and ruling out the safety issue of using viral protein. So the last module is what we call the targeting system. And here we develop a very versatile system that really enables to recruit at the surface of the vesicle any type of validated biotinylated antibodies that can be for R&D grade or clinically approved. So the idea here is to recruit the antibody to target the cell toward the cell of interest that will harbor at the surface an antigen of interest. And as you can see here on the graph, you can see that the targeting is very efficient, and we can very precisely target, in that case here, CD8 positive cells. And you see a nice dependency of the dose when we increase the amount of CD8 positive cells. So now when we assemble these three modules, we decided to develop new application, because we are very early stage, we call that application, but hopefully in the near future, they might become products. But we need time for that and partners. So the first application that we targeted is killer EVs. So basically our goal was to develop killer EVs to eliminate tumor in the future. And so here we took advantage of our technology, and basically we loaded a very lethal toxin inside the vesicle to kill the acceptor cells. The second application that we developed are so-called editing EV. So here the goal is to load the editing machinery, Cas9 CRISPR, and basically be able to reprogram the cell of interest. And in our case here, we could already reprogram CD8 positive cells. Just some example with a more mature application, the killer EV application. So we completely validated the efficiency of the killer EVs in vitro. And here you have an example from our paper, when you can see in the orange histograms that very efficiently we can almost completely kill the acceptor cells. And now we move forward in vivo studies, and we just started to do in vivo validation by testing anti-tumor activities of the killer EV in mice bearing a human tumor. And as you can see here, we can see a reduction of the tumor progression, so that's the blue curve, and when we do multiple injection of EVs. So that's a work in progress, and of course we need to continue to see if we can stabilize and completely inhibit the tumor growth. But I think it's promising. So that's not the only activity. So what I presented here is basically what we believe will be the generation 1 of EV-based vector. But we believe that what needs to be accomplished to be even more efficient and close to the biology is to really discover the key protein that control the endogenous uptake of vesicles. So for that, our goal is to discover the key machinery that control the uptake and the control delivery. And for that, we are performing a genetic approach with a unique library that we call surfaceome, because we are targeting all the proteins that are at the entry point of the vesicles. And hopefully in the upcoming years, we will know what are the endogenous proteins that are specifically involved in the uptake, and more importantly, in the fusion, to position us in a unique place to have a new generation of physiologically relevant EVs. So we presented a new technology that presented several advantages that I discussed already. Obviously, we're at a very early stage, and we need to establish partnership with a scientific partner and also sponsors to, at someday, hopefully position the EV-based vector in existing pipelines and in new pipelines that will hopefully enable to reprogram cell in vivo. Thank you for your attention.
Thank you. Yes, and then you'll come back for the questions at the end. Thank you very much. So now, Jérémy Laurent, okay, you're going to present your company Astraveus, and you're going to talk about microfluidics associated to cell and gene therapies.
Hello, everybody. So thanks a lot for the organizers for inviting me to present Astraveus. And thanks a lot for giving your attention that time of the day on the Friday as well. So I'm going to speak about Astraveus. We are a startup created in 2016, spinning out of the St. Louis Hospital, the laboratory of Jérôme Larguéron, more precisely. So we define ourselves as the benchtop cell factory company. Our goal is to take away as much as possible of the complexity of developing and scaling the manufacturing of cell and gene therapies, so that all the brilliant scientists that we have heard today can focus more on the science that brings innovations and move us past the current frontiers in cell-based therapeutics. So we've had a number of introductions already on the space of cell therapy. So I think the potential is quite clear in the audience, and everybody knows as well as of the problem for resource intensivity behind the manufacturing of those very complex drug products. So the second, the lower part of the slide, which depicts the difficulty of scaling the manufacturing, I think is a well-known problem, where it takes a lot of clean rooms, people, instruments, which ultimately is just something very complicated to do at the right scale, and also takes a lot of time to do. With the situation today that taking the approved product, the fraction of patients that could benefit and actually do is quite small, around 2% currently. There is also, in our opinion, a bigger, or at least as big part of the problem on the research and development, or more precisely process development side, which is how we get this right. Because we are talking about every time we do an attempt of making this type of complex product, using a lot of resources, easily spending 50 to 70,000 euros, using amounts of patient or donor materials which are ultimately limited. All of this for poorly controlled procedures, often quite manual, for only one test. So basically optimization is kind of infeasible today. And given the complexity of the drug product, this is really a missed opportunity to make better in the clinic later. So these are the problems that we are tackling, and the angle that we are taking at Astravus is first and foremost a radical miniaturization. Our view is really miniaturizing a conventional cell and gene therapy factory into a benchtop system. So since 2016, when we've started the work, we've been developing technology to make it into a microfluidic system, so to bring the end-to-end manufacturing of cell therapy product into a microfluidic unit, which you see on the left of the slide. It's a book-size system. And note that we have this system with a one patient capacity in terms of cell number, depicting also higher precision and higher efficiency thanks to the intrinsic benefits of microfluidics. We are able to build the system that you see on the right. So our first benchtop cell factory, which is then capable to make therapies for 10 patients, and is fully automated, so really minimizes the number of manual operations, including some of the current QC bottlenecks. So you can see there's a microscope inside the system, and recently we've been able to show that we can recapitulate the equivalent of flow cytometry in terms of quality control. So our work is pretty advanced, and we hope to demonstrate that we can address the manufacturing problem that I mentioned earlier, and especially that we will enable the current teams and the current manufacturing facilities to make therapies for way more patients, and that way brings these benefits, including in liquid oncology, to way more patients. But we also hope that the tool that we are developing will be leveraging innovation in the space and help bring better cell and gene therapy products in the clinic faster by essentially streamlining the process and the process development, making the researchers able to run tens of experiments repeatedly in a very automated manner, including deep readouts with these analytic modalities that I quickly mentioned. So this is the wrap-up. Sorry, I went a little bit past the time. So we are building this platform to simplify the life of innovators in the cell and gene therapy space. The key items of our value proposition are simplification, speed or throughput, and bringing cost and environmental impact reduction to the whole process in R&D as well as in manufacturing. Thanks a lot for your attention, and I welcome the discussion.
Last short talk. Now, Jérémy Bonnet, this time. Okay, two Jérémys, one afterward the other. Programming bacteria as smart therapeutics. You're from INSOM.
All right, thank you for the introduction. Hello, everyone. Thanks for the invitation. It's been a great pleasure to be here these 2 days and see these incredible advances in genes and cell therapies. And so my name is Jérôme Bonnet. I'm INSEM researcher. I work in Montpellier at the CBS. And we are a team of 15 people dedicated in engineering bacteria to improve health. So we've seen a lot of cell therapies and incredible advances. But actually, there's one type of cell that we haven't talked much in these 2 days, and which are actually also cells. These are bacteria. And as you know, bacteria actually play key roles in our health, and for that, they're real interesting therapeutic agents that we want to use to actually improve human health. So we can use engineered bacteria actually to modify them to make them express different kind of genes, like antibodies or enzymes. And we know that natural bacteria can actually modulate our metabolism, our immunity, even our moods. And basically, there's even already some proof in animal proof of concept, of application to metabolic disease, infectious disease, and cancer. So this is very exciting, but actually, if you look at the first generation of engineering bacteria, you look at they basically constitutively producing a drug. And so it's like you don't have really control of them. You cannot activate them from outside. They don't know where they are in the body, and they cannot detect the place of disease. So if you are driving a car, it would be like driving blindfolded and full speed, which usually leads up to that. All right, so now with the tool of SynBio, we can actually engineer these cells and give them, in addition to weapons, we give them eyes and brains. And so what we want to do is engineer sensing and computing modules to make them smarter. And by doing that, we hope to make them more efficient and more specific, safer, more reliable, and importantly also reconfigurable, which means that basically we want to use modules that we design in a plug-and-play manner to readapt a particular chassis to many therapies. So if you keep the car analogy, basically now it looks smart bacteria more like this. All right, so what are we doing? First, to engineer sensing, we engineer receptors that actually can detect different signals. And here we use a synthetic receptor platform, which is modular, in which DNA binding domain activate transcription. And so the idea here, which is important, is actually to be able to exchange the ligand binding domain only to detect new ligands. And in this way, you can reprogram detection. So we did that for physiological signals, for example, bile acids, which are a marker of intestinal location, but also a marker of liver dysfunction in the serum. And you can see here, the bacteria can actually detect them and produce purple color when they're in the serum of a patient with liver disease. We changed this domain for a domain that detects caffeine, and now the same receptor can actually detect caffeine. You can see the fluorescence of the cells actually increasing here, so detecting an exogenous signal in that case. So now we give them eyes. How do we give them a brain? Where we inspire from computing and electronics to engineer these tiny DNA switches that can actually be flipped and have two states, like 0 and 1 in computer science. And here, when you flip the switch using a recombinase, you can actually trigger gene expression. And this is reversible, so now you have a memory that remains for many generations. You can imagine have sustained drug delivery once you activate your device. Also, we can combine these switches to engineer logic gates. So in this case, an AND gate, where you have production of your gene only when the two signals are present. So the signal A and B, you can see here the bacteria producing GFP. Okay, so these are the tools. Now I want to show you how we apply them for therapeutic bacteria. So yeah, I show you some work, we're actually ongoing work, where we are engineering bacterial cancer therapeutics, all right? So basically, you can imagine now that we know that bacteria can colonize tumors, and you can imagine that by giving them coffee, your doctor could actually activate therapeutic release at the right time and the right dosage. So here we take our caffeine receptor and you replace GFP reporter by luciferase to visualize its activity in vivo. And then we inject tumor in mice. And then once the tumor grows, we inject bacteria that colonize the tumor. And then we give them caffeine by gavage. And we monitor luminescence. And by doing so, we can see that when we give caffeine to the mice, we can actually observe luminescence, which means we're able to control gene expression in vivo in bacteria. So we're really excited about that because this is remote control of cell therapy in vivo. And now we're pushing that towards therapeutic effectors currently in this actual moment. Okay, so now I gave you a view of how we want to program bacteria by using programmable receptor to change ligand detection, using computing and logic devices and memory to allow them to process signals. And I told you also that bacteria are actually really good at making protein, for example, enzyme, VHH, but they're also metabolic workhorses. And we're really excited about using this metabolic cluster to degrade or actually synthesize complex molecules in situ and metabolically change the metabolism actually locally or even globally in the body to actually have different effects. And we can also imagine have a direct therapeutic effect with that, or even potentiate other therapeutics in complementing them. What could be also cool is to actually be able to visualize these cells in vivo and see where they are and how they grow. And to do that, we use this tiny gas vesicles that are agent contracts, so self-assembling protein that can encapsulate gas and that you can visualize with ultrasounds. And
in this case, we're also working on that kind of stuff. So what I saw in these days is even towards cell therapies, what's really exciting for us is synergizing. Here I want to show an example of a synergy between CAR T cells and bacteria. So here's a Danone group in Colombia actually use tumor colonizing properties of bacteria to then release specific antigen recognized by modified T cells along with chemokines to attract them to the tumor. And I think it's a really good example of how you can combine these therapeutics. And I believe there's a lot of different examples that we can actually find and combine this therapy together. Obviously, there are also many challenges. So obviously, we want to be better at manufacturing these bacteria, even if they are actually very robust. We need good processes to have reliable production and good quality. We need also to be able to contain them to avoid their escape into the environment. And obviously, we need clinical validation. We're not here yet, so the road is still bumpy. But we need to get better at get a better efficiency in vivo and understand how to actually deal with these cells and the interaction with the body. So with this, I would like to thank my team and all our collaborators and also all the founders that support our work. And then I thank you for your attention, and I'll be happy to discuss later.
Thank you. You can stay on stage. Thank you. And then Grégory Lavieux and Jérémy Laurent, you can come back for a few questions and answers. Okay, so do you have any questions?
Yes.
Yes.
So to the last speaker, please. Could you tell us a little bit more about how these bacteria go to the tumor specifically, or what kind of bacteria? Why would they not be eaten up by macrophages? Or
yes. So we don't actually know. That would be the answer. That is the more appropriate answer. So this has been known for a long time, like since Dr. Collins in the 19th century actually found that patients got infections in their tumors, and he observed tumor regression and believed that actually triggers the immune system, so the first immunotherapy. But it's not really clear. Like people thought it was EPR effects, but actually there's some the vascularization is permeable, so it allows bacteria to go through all. But if you really look at that, I doubt it. So I think the data is not clear on that. It will be really interesting to study actually how do they colonize a tumor. Then they get wiped out from the body. So if you give that to people, for example, you can inject millions of bacteria. They're going to accumulate also in the liver and the spleen. But over a few days, the immune system, we're going to keep them out. And I believe the immunosuppressive environment of the tumor might help actually not getting rid of them, and then they can grow, because you need a few cells to then grow. And first, people were using pathogens, but now we're trying to actually use probiotics. So like what I show you is E. coli Nisel 9017, which is a common probiotics.
Thank you. Other question? Yes, Marc?
So it's a question to Jérémy Laurent. I was wondering what kind of cells can be grown in your system?
Thanks for the question. So right now we are really focused on CAR-T for obvious reasons. The technology is really ready to produce any type of non-adherent cells, but the technology is amenable to adherent. Essentially, we've got the building blocks for both adherent and non-adherent culture, as well as enrichment and other process steps. So the thing is just, we are already 43 people, which is, great, but those machines are a little bit like planes. They take a lot of resources to build and a lot of people to operate today. So we have to focus, and for obvious reasons, for the moment, we focus on CAR-T.
And basically, so how does it work? I mean, you can maintain, so since it's a closed system, so you inject, I guess, your inoculum, you add some growth factors and so, but you have continuous feeding, you have retro control of the concentration and all this stuff, no?
Yeah, that's many questions in one. So generally speaking, the way we look at it, we really want to miniaturize the number of manual interventions. So with this modularity that we have, you can, for example, mutualize the input of all the reactants, at least the ones that are not extremely temperature sensitive. So all of those can be connected at once. Then if it's an autologous product, you would need indeed to have different inoculi, whether simultaneously or asynchronously, depending if you start all manufacturing for different patients at the same time or not, as the platform is amenable to scale out strategies for allogenic as well. Yeah, and then you'd have additional manual interventions, obviously, for product recovery, as well as sample outtake for offline analysis that are not performed by the system. And then in terms of programmability, we can really play a lot with the system. The basic operations that you would do either in an artisanal process or with another type of integrated bioreactor you can do. There are sensors, so there is a lot of embedded flexibility. And for example, as you suggested, you can build in feedback control loops for perfusion culture. That's obviously one of the possibilities with the sensors or the different analytical capabilities. And it's all the point of having the same system capable of doing process development as well as scale-out manufacturing, is that you can really use this modularity in the process development stage to develop elaborated processes that build better products and better efficiency as well, and then not do any transfer or scale-up at all, actually do a scale-out. You already have your clinical manufacturing process that you don't need to change ever again. That's the whole point of having the same system for both types of work.
Thank you. Yes.
Yes, I was very impressed by your talk, Jérémy Bonnet, about all the potential you have with those bugs. So I was thinking, because I guess for treating solid tumor, we have to be local for the treatment, and indeed it's very important that your bugs colonize in the tumor type. But you could also, in addition to this logic gate system, think about having a trap system. For instance, IL-10 is produce an immunosuppressive environment. So if you have a way to give the bugs the capacity to eat the IL-10 and then to trigger indeed a response, that could be a way also to have sensor for hypoxia or acidity. I guess you have already thought about that. So that would be really cool. But indeed, again, the same question as before, you have to be really sure that your bugs are going to stay there and not act somewhere else in the body.
So to the end of your question, thanks, I would say, well, today, when we first tried this therapy, like some people were actually, cured, and some people were dying from, infection and stuff. But it was before the invention of antibiotics. So I think all the things you go to human with these bugs, first of all, they're not resistant to antibiotics. So if you have a problem, you can actually take antibiotic, and also engineering kill switches to, destroy them on demand. What you can think about also is confine their growth to particular environment. So like you talk acidic thing, like lactate, for example, that kind of things. We have sensor for that, and we can actually connect that to genes that control, essential pathways. So you have an oxotroph who can grow only in a particular place. And obviously, what you're talking about, modifying the tumor microenvironment, well, obviously, I'm not an oncologist, right? So I need help with that from colleagues. But it's definitely things that we are trying to do, either with chemokines. We can also secrete immune checkpoint blockade nanobodies and single domain antibodies, and that kind of things. So yes.
Thank you. Yes. Can you introduce yourself? I forget every time, sorry.
Yeah, sure. So it's Erminia Rubino from Institut Imagine. So I had a question from Gregory. So the approach of using syncytin like to improve the fusion of some particles is quite interesting. Other teams have shown that there are possibilities in that field. So I have a question about the immunogenicity of these kind of proteins, because I guess the dream is like using endogenous protein would reduce probably immunogenicity of fusogens. instead of using viral origin fusor genes. So that would be the first point. And the second point would be more like the drawback of using these endogenous proteins, since we know that they have other physiological roles. Do you see in your, especially in the in vivo experiments, some drawback of these types of off-targets of the technology due to the natural functions of this protein?
Thank you for the question. So the first part concerning the immunogenicity, so I think that because the human protein, there is a real advantage if we do the straightforward comparison with viral protein, for instance, VSVG. So I think that's a real plus. And I think it's going to address something very important for the immunogenicity by strongly reducing it. So the second part, the off-target effect, so that might happen. As you see, we are using several modules. And so what we know from the in vitro validation is when we use the targeting system, we basically outcompete syncytin-1, for instance, right? So we believe by doing that, we'll be able to really direct the vesicle towards the cell of interest. The second thing is that so there is syncytin-1, but you have also syncytin-2, for instance, right? And the distribution of the receptor is not ubiquitous in all the cell type. So depending on the application, you might have a better choice for one particular syncytin or another ERV protein, so the same extended family, to target cells that have the cognate receptor for that protein and take advantage of the physiology in that situation. So it will depend on the application.
Thank you. Other questions? Yes, please. Introduce yourself.
Yep. So Gavin Badamale from Art Design. Question to Gregory. So you mentioned one of the strategy for targeting is immunoconjugates, so that you bind the EV to an antibody. And I was wondering, is the EV population homogeneous in size? That's the first question. And the second one is, what would be your priority in terms of therapeutic areas, meaning where would you test that first?
So in terms of size, right now we are agnostic of a particular subtype, and so what we use is it's a genetic tools, right, to position our chimeric protein that are massively overexpressed. So we believe, and the results suggest that most of the vesicle will be equipped with copies of the chimeric protein that will serve what we call the adapter protein, right? So what we are not sure is, do we have the same number of copies for each vesicle? That we don't know. We did not address that. So I cannot guarantee that they will have the same number of copies, but what we know is that most of the vesicles will be equipped with the adapter.
But then if the size is heterogeneous, the payload will be heterogeneous also, right?
The what?
The payload, if the size is different?
Well, I don't think, I'm not sure that the size matters that much.
And then about the priority areas for therapeutic, which indications would you want to test?
So right now what we have identified for our own application are, CD8 for the editing EVs, because we believe we can make a major impact if we can reprogram CD8 positive cells. So the second application with the killing activity, we would like to, as for the proof of concept, to target HER2 tumor cells, which is the examples that I show. For the rest, we are really open. What we really wanted was to have a versatile approach, to have something that is strong and robust, so then we can basically change the targets when needed.
Thank you. Okay, thank you very much. So now it's time for the final keynote, and we're very pleased to welcome Professor Alain Fischer. I think, Professor, we're going to commemorate this year the 25th anniversary of the release of your discovery and, of course, the Bubble Babies gene therapy, which was a world first. And we're happy to welcome you for this final note. I remind that you're a professor at Collège de France and president of the French Academy of Sciences.
Yeah, now it works. Thank you. So thanks for the invitation, thanks for the introduction. So I was not there for the whole 2 days, but I heard this afternoon some interesting talks about basically cell therapy, and obviously altogether you are working in a dynamic field, and I think the timing of this conference was certainly well taken. Maybe the very first remark I would like to make, which is an obvious one, but I think from time to time it's good to repeat it. That is, if you look at where the advances came from, they come from basic science. We should not forget that. I mean, think about CRISPR-Cas9, think about siRNA, think about checkpoints of immune responses, and so on. They were all discoveries from basic scientists, and so this is important to remember, and for politicians, this is important to tell them that to support basic science is probably the best way to get, at the end, new drugs and advances in medicine. Since I know better gene therapy than any other field, I will focus most of my remarks on gene therapy, although I think some of them may be applicable to cell therapy or RNA therapy too. Maybe an interesting observation to make today, I mean, with the advances in the field, is that we are shifting a little bit from rare diseases to more common diseases, which I think is, of course, extremely significant for the future. But let's briefly examine what has been done in gene therapy. The most obvious important development is the one with CAR T cells, with the use of synthetic genes, of course, as you know, and this is a major advance over the last 10 years to treat a variety of B-cell malignancies. I don't need to go into detail, but as you know, there are 6 drugs today that have been commercialized, and I think worldwide more than 20,000 patients have been treated. In France, I think we are close to 1,000 a year, which is quite significant and raises a number of questions. I will come back to these questions at the end of my talk. And obviously, there are many perspectives in the field of CAR T-cell therapy, of course, to increase efficacy, to reduce toxicity, which is still an issue. Of course, the next challenge is to make them useful to treat some solid tumors. I think there are a little bit of advances there, but this is not yet achieved. And I think, and this is very exciting, and I think you talked this morning, if I'm correct, about the development of CAR T cells to treat autoimmune diseases, at least B-cell antibody-mediated autoimmune diseases. I think this is extremely exciting, and I'm pretty convinced that this will be a field of development in the next years to come. Then comes the treatment of genetic diseases. Today, in the last 25 years or so, approximately 20 inherited monogenic diseases have been treated with significant clinical benefit for patients, not always a cure, but at least a benefit. So in the field of ex vivo gene therapy with lentiviral vectors, this includes, I think now 7 primary immunodeficiency diseases, so some metabolic diseases, some of them very difficult to treat, like metachromatic leukodystrophy or mucopolysaccharidosis, or then the advances are extremely dramatic. And of course, also the treatment of sickle cell disease and beta thalassemia, where classic gene addition by lentiviral vectors have had some effect. And of course, then comes the in vivo gene therapy with AAVs. As you know, significant results have been obtained in the treatment of hemophilias, of liver disease, of spinal muscular atrophy, and this is quite spectacular. But there are still quite a number of limitations, despite this significant number of successes. One is manufacturing. I mean, we hear just now some interesting talk about how to manufacture cells or vectors. And I think the issue of production of vectors, and for AAVs we need usually large quantities, remains a difficulty, as well as when the vector is prepared for ex vivo gene transfer, the ex vivo transduction manipulation or manufacturing is still quite cumbersome, and if it can be simplified, it will be an advance. And we need, obviously, to go to automation
And I think the last talk was on this prospect significant. Another topic of interest where there are still limitations, if you think about ex vivo gene transfer and infusion into patients of stem cells or T lymphocytes, then you need the selection process, and this is usually done with chemotherapy, which can be done, but obviously which can be toxic, and probably there are some tricks, especially in the field of hematopoietic stem cells, may be used in the future to avoid to use any more chemotherapy, but rather, for instance, antibody or molecules that will attract the stem cells into the niches, into the bone marrow. With AAVs, there are some quite significant issues. I mentioned the need for, at least for neuromuscular diseases, to use quite a high dosage of vector, and this is a problem because of toxicity. I mean, this has been clearly shown in a recent past, where there are two problems, which is immediate toxicity caused by innate immune response that can be dramatic, and some patients died from it. And then secondarily, there is the immunogenicity of the vectors that play a role in limiting reutilization of such vectors. So these issues, and I think this has been a little bit underestimated over the past years, but today I think the questions of immune responses altogether, both innate and adaptive to AAVs, have to be tackled for future development. But of course, then comes the new steps, I would say, the second stage of gene therapy with genome editing, and I'm sure, of course, you discussed it, and based on the CRISPR-Cas9 system. And of course, the system per se, as you know, can be used to inactivate genes or regulatory elements of DNA, and this has been used with a spectacular success to the treatment of sickle cell disease and beta thalassemia, and there is now a drug approved that can be used to reinduce expression of fetal hemoglobin. This is an extremely elegant approach, which looks quite good. And some other examples come with the downregulation of kallikrein in patients with onconeurotic edema. And maybe this is a field where the more frequent diseases will be tackled, like hypercholesterolemia, because if you reduce the expression of PCSK9, this molecule that is involved in the metabolism of the bad cholesterol, you may control hypercholesterolemia, which is, of course, not a rare disease. But as you know very well, the CRISPR-Cas9 system as such cannot be used in other instances because it induces a number of targets. So, but there is now the second generation of genome editing that is coming, that has already been used in patients with several approaches. The base editing, which is the first one with the modification of nucleotides based on usage of deaminases associated with NIC Cas9, so preventing the development of double-strand breaks into DNA, which are causing the problems related to CRISPR-Cas9. But beyond base editing, there is prime editing, where you can introduce into the DNA some stretch of a DNA sequence based on adding a reverse transcriptase into the guided RNA. And further developments may come for even more subtle or sophisticated strategy like paste editing with an integrase, or even transposon addition to introduce a full gene or quite a long sequence of DNA into the genome. And there are pioneers like David Liu at MIT that are developing these strategies that I think are extremely interesting and will lead to a number of clinical applications beyond base editing. But delivery is another important aspect, delivery of the genome editing products, but also the delivery, as I mentioned, of more classical DNA approaches. But if we can get rid of viral vectors, and I mentioned the problem with AAV, this would be fantastic. And this was mentioned earlier this afternoon, the usage of nanoparticles, lipid nanoparticles with ionosable lipids is of major interest, provided that I mean, for some of the applications where there is a need to target the particles to the cell of interest, this could be achieved by adding on at the surface of the lipid particles an antibody or a protein that will target the particle to the cell. I mean, there has been recently a publication about targeting hematopoietic stem cells in mice that looks promising, but we need to see further development. So there is a need to fund research. There are plenty of opportunities, a lot of questions to be solved, but again, with many potential applications. So it's great that this is being developed today in France with the, at least to support bioproduction, but not only bioproduction should be supported. I think research prior to go to the stage of bioproduction has to be quite significantly supported in order to be innovative and really to rise, to develop new products. Okay, but then I'd like to finish by developing a question which I think is an issue about the future development of cell therapy, gene therapy, RNA therapy, whatever, these new biotherapies, which is the price of these innovative treatments. I'm sure everyone is aware that all of these drugs, those that have been now on the market, I think there are 16 or 17 in the field of gene therapy, they have all a very, very high price. So the cheapest, if I may say so, are the CAR T cells with approximately 350,000 euros, but all others gene therapy are these days in the order of magnitude of 2 million euros, which is extremely expensive and by far the most expensive drugs that are on the market. So this is based on grounds that are, to my opinion, debatable. The three grounds are, one, these are sophisticated products. The second one is that usually the market is small. And the third one is that these are transformative therapies. But I would like to argue that, okay, this is more sophisticated usually than classical chemicals. But in most of the cases, if not all for gene therapy, the products have been conceived and tested up to the first clinical trials in academia or with the help of charities, so that the pharmas then, whatever they were, first startup and then bigger pharmas, they are reducing the risk by buying a product that has been quite advanced already. The size of the market does not seem to influence the price, so I don't think this is an acceptable argument. And the last one is transformative therapy. This is something of importance. Nevertheless, one should consider the fact that for most of these diseases treated up to now, whatever these are, cancer or genetic diseases, this is not always a cure. I mean, patients still require quite a number of treatments and care. So this is not all or nothing. And especially there are sequelae of neuromuscular diseases, for instance, and so on. And even if it would be the case, maybe the question should be raised, if there is a benefit, should the benefits go all to the pharma, or should it be shared in a reasonable way between the pharma and the society? I think this is an important question. So there are consequences of this. In the US, there is, as you know, very little regulation of prices, so that the prices are extremely high. In Europe, it is not so. This is not so, but still, the pressure comes from the US. But it leads to the fact that, for instance, one company, Bluebird, involved in the development of gene therapy products, has decided not to come to the European market because they consider that the prices that they will get is not high enough. Of course, this is a problem in terms of accessibility of these drugs for patients. So what can be done? Is it possible? Is it feasible to reduce prices? Well, technically speaking, I would say there are some points to be discussed. First of all, with time, we may hope that the prices of a given drug will go down, although there are examples in both biotherapy where this is not the case, if I can develop if you wish. We can hope that automation and maybe to regulatory processes that are slightly less constraining may lead to price reduction, let's hope. But this is not enough. And I think we have also to really consider the economical issue. And then I would like to briefly discuss, I am at the end of my time, the Barcelona case. I mean, probably you know that in one hospital in Barcelona, the people there, they have developed a facility to make their own CAR T cells that are delivered to patients, and the price at which the product is paid by the Social Security in Spain is 90,000 euros. So that is 3 to 4 times less than the prices from the CAR T cells developed by companies. So this is a priority medicine designation system that works in Spain. So it means, I mean, these simple examples shows that it is possible to pay less for a given sophisticated product. And we have to work on that. We have to work on that, I guess, by duplicating, developing such initiatives like the one in Barcelona. And I know there are some attempts to do it in other places in Europe. And I think we need to have not-for-profit production units of course, GMP grade drugs for gene therapy. This is absolutely required for the treatment of ultra-rare diseases, but perhaps also for less rare diseases. So I'm very much in favor of thinking about how to put together investments from the public as well as foundations and charities to develop such units. I think another aspect, and we're almost done, is that instead of having price negotiations at the national level, and I know that in France they do what they can within the CEPS, but if it will be done, for instance, at the European level, associated with EMA, I guess the strength in negotiation with pharmas will be stronger and cheaper prices can be achieved. I mean, there is an example of the feasibility of that approach. There are the vaccines against COVID. They were negotiated at the level of the European Union, and it works extremely well. So I think this is not easy, of course, to organize, politically speaking, but I think this is a way to go. And perhaps, by the way, more transparency, worldwide negotiation, that's, of course, very much more difficult with pharmas, might also lead to some reduction in prices. But in any case, I think it becomes urgent, given the fantastic opportunities that are developing in these different fields of biotherapies, to tackle this issue of prices if we want these medical innovations to profit everyone, at least in developed countries. Thank you for your attention.
Thank you very much, Professor. Thanks a lot for this conclusion. And now the final conclusion after this very important speech. Thanks a lot again. With Marc Bonneville, chairman of ARIS, you could see during these 2 days that he was very much implied with many questions, pertinent questions to help and to understand better. So now it's your time for conclusion.
So I'm surprised we are more than 10 at the end of this meeting, which is a good sign. And I thank you all for staying so long. Clearly, from the standpoint of ARIS and hopefully from the standpoint of the other organizers, I think that the meeting has fulfilled our expectations. Namely, it sparked very stimulating and constructive discussions between both naturally public and private players. And hopefully it helped establish potential partnerships through the many B2B meetings that have been organized. Basically, we have filled most of the slots that were planned. So the meeting was also a great opportunity to highlight the excellence of the French researchers and innovators in the field of cell and gene therapies, thanks to the excellent overviews and pitches on upstream and downstream topics, thanks to the very active roundtable discussions as well as presentations of several assets of the French ecosystem that make it particularly conducive to the implementation of cell and gene therapies. So I won't have time to summarize here all the topics that have been discussed, but we'll mention a few bullet points. So regarding AAV and lentiviral therapies, as well as gene replacement or editing approaches, several excellent centers in France gathering translational expertise covering all the aspects of product development
have been presented. Several shared challenges regarding costs, specificity, efficacy, toxicity, immunogenicity, lack of relevant preclinical models, and with a recurrent issue regarding cell and tissue targeting, as well as toxicity associated in particular with off-target effect of some gene editing technologies, have been extensively discussed, and I think in a very productive and constructive fashion. There have been also a number of promising perspectives that have been highlighted, such as improved AAV vectors through chemical modification, computational design, and through harnessing of AI-generated libraries. Also, implementation of DSB-free gene editing approaches with clear opportunities to increase potency and specificity, decrease doses and toxicities, as well as manufacturing costs. Regarding mRNA-based clinical application, these were nicely reviewed by Chantal Pichon, and the ensuing discussion on siRNA and oligonucleotide strategies, in particularly during the roundtable of this session, highlighted not only several recent successful achievements with mRNA vaccines and siRNA, but also here again recurrent challenges dealing in particular with specificity and targeting, as well as opportunities to better leverage AI/ML-based approaches to improve design of the vectors and these approaches. Regarding immune cell-based therapies, mainly CAR T cells, so these, as you know, have yielded many promising clinical results in both oncology and more recently in autoimmune diseases. But there are here again many challenges and unmet needs. In particular, how can we apply this approach to tackle solid tumors? How could we use allogeneic CAR T cells to reduce costs, improve standardization, and scaling up? Many other issues were largely discussed during the roundtable regarding efficacy, which depends on the definition of the right target antigen, the control of tissue homing, survival, effector function, ability to thwart tumor escape mechanism, as well as toxicity, natural toxicity issues. So given the diversity of unsolved issues, all the participants, and I think also the audience, agreed on the importance to involve all relevant experts from both the academic and private side, but also patients and regulatory players to move forward efficiently. Finally, regarding non-immune cell-based therapies, so while promising clinical data have been obtained here again, discussion focused on the actual mode of action, in particular of MSCs, seemingly primarily through supportive or endocrine activity rather than regeneration. Also, so there was discussed the need for robust qualifying and potency assays and the respective caveats and advantages of cell versus exosome-based therapies. An important point that was raised was the importance to follow up the patients and share data, clinical and real world, ideally, for the benefit of the whole community to accelerate cell and gene therapy development and allow accurate assessment of the cost-benefit ratio of these approaches. So just to finish, I would like to, and most importantly, acknowledge a number of people who have been key in the organization of this meeting. So naturally, well, Stéphanie, Véronique, and Jean-Pierre from Aris, Violaine Gauthier and France Biotech, Nathalie Manot and Delem. Nathalie did really a fantastic job. Thank you, Nathalie. Rosalie Maurice, Julie Lelouch, and BPI France. Franck Le Timonier, thanks a lot, Franck. You were instrumental in the success of this meeting, and as well as other ITMO directors and the whole INSERM teams. Jean-Marc Gauthier, Hervé Coste, and Gloria Baton from INSERM Transfer were of strong support throughout the organization of the meeting. Several students from Sud Biotech, Amanda, Matteo, Océane, and Yasmina. Fabrice Papillon, thanks Fabrice. Thank you. It was a very dynamic discussion. People were impressed and thought that you were a scientist. You are a scientist almost.
No, but I could turn into.
Yeah. I would like also to thank on behalf of the organizers all the academic, biotech, and pharma speakers. You did fantastic presentation, fantastic discussion, and naturally all the RT roundtable participants. We would like also to thank the excellent, the AIS and SGPI, and in particular Lise Alter and Bruno Baudel, who did a really nice presentation of the French ecosystem, as well as the Ministry of Economy and Finances, who kindly hosted the meeting and in a fantastic environment. So thank you very much. Have a good trip back and hopefully establish many, many fruitful collaborations. Thank you.